pick up here with objective number 4. So in our first packet, we really spent some time describing these structures, describing these endocrine glands and organs, and really kind of laying the groundwork, speaking about that hierarchy of the endocrine axis, of the hypothalamus, anterior-pituitary endocrine gland axis, and so in this packet, we're going to get into the details of 1 or 2 of those systems. So specifically the thyroid gland and how thyroid hormone works. We're also going to speak a little bit about the adrenals and some of the layers of the adrenal cortex and some of the hormones there. We're not going to speak at length about the pancreas because we'll talk about it in the GI system, so in the gastrointestinal system. We'll talk a little bit about it later on. And that's kind of all that we will say as far as the endocrine glands, right? The sex glands, so the gonads, we'll talk more about them next semester in the reproductive system. And so the ones that we're really going to pay attention to are really just the thyroid, the adrenals, and then a little bit on the pancreas, okay? So there are other endocrine glands that are involved that we drew in that axis. We're not going to say much about those, but we will talk about the ones I mentioned here. So objective 4 is to describe the synthesis and secretion of thyroid hormones, to distinguish between direct and permissive actions of thyroid hormones. So looking at the thyroid gland, it is located in the neck region. So it's anterior and around the trachea. This is how it looks here. So just below the larynx. It secretes 2 important hormones, 2 thyroid hormones. There's T4, tetraiodothyronine, also called thyroxine, and T3, triiodothyronine. And these are really important as far as regulating our metabolism. And when I say metabolism, I mean the rate at which our cells are using energy and are actively functioning, okay? So the rate of cellular function. The thyroid also produces calcitonin, and calcitonin is what helps to regulate calcium levels in the blood. So 2 somewhat separate functions from these hormones that are being released from the thyroid gland. And the cells that secrete thyroid hormone are entirely different from the cells that secrete calcitonin. So let's look at the functional units of the thyroid gland. This is called the follicle. And lining that follicle, there are cells called the follicular cells. And just for reference, these are the cells that create thyroid hormone, so T3 and T4. The cells that create calcitonin are the other cells that are in between our follicles. All right, so just to kind of distinguish the groups of cells that are creating thyroid hormone versus the cells that are sort of interspersed in between the follicles that are creating calcitonin. So the follicular cells are the cells that are using what's called colloid to make this thyroid hormone. So until thyroid hormone is ready to be made and ready to be circulated in the blood, it is stored within the follicle as colloid, okay? And colloid is really just thyroglobulin. It's a precursor protein. It's that storage form of thyroid hormone, and that's what makes up the central aspect, this yellow region of our follicle. Now, when it's time to release that thyroid hormone into the blood, remember endocrine hormones must be released into the blood. What we do is we use iodide, and iodide is the ionized form of iodine. So iodine is what you take in your diet. And when we ingest that iodine, it can be activated into an ion that can be used to transport or activate that colloid and make the thyroid hormone. Okay, it's going to be added to that thyroglobulin to make thyroid hormone that can be circulated by these follicular cells into the blood, and then that can be sent around our body as thyroid hormone. Okay, and that's why it's so important to have iodine in the diet. If we have a deficiency in iodine, if we're not getting enough, or if we have a deficiency in using it appropriately or in making thyroid hormone, what happens is these follicles start generating more and more and more of that storage form of thyroid hormone, that colloid, and this is what gives us that goiter. So when the follicles eventually get larger and larger and larger, because they have all of this precursor colloids stored up in here, all of this unactivated thyroid hormone, and there's no way to activate it because either we don't have the right resources or we don't have the iodine to do that appropriately. We end up with a large swelling of the thyroid gland called a goiter. Okay, so that's just basically swelling up of the colloid. All right, so let's look at the mechanism by which we can take this colloid and make it into thyroid hormone to be circulated in the blood. So essentially, we have to start with TSH. And if you remember that axis that we spoke about, TSH, also called thyroid stimulating hormone, is being secreted from the anterior pituitary. And just to go a step back further, it is being secreted in response to TRH, thyroid releasing hormone, which is coming from the hypothalamus. And so when we have TSH coming from the anterior pituitary, circulating in the blood, it's going to arrive here at the follicular cell of the follicle of the thyroid gland. It's going to bind to a TSH receptor, so a very specific receptor for TSH, and then it's going to bring about some cellular response. Now, this process should look very familiar because this is a metabotropic response. So this receptor is coupled to a G protein. It's going to have part of that protein dissociate off. It's going to activate some amplifier enzyme downstream. That is going to generate second messenger, which is cyclic AMP. That's going to turn on protein kinase. Protein kinase will phosphorylate some protein, and that will help to generate the reabsorption. So a large chunk of that colloid is going to be reabsorbed, and we're going to be able to turn that into thyroid hormone and then secrete it from the cell. Okay. Now, if we look more closely at what's happening within the colloid, within that storage form, that storage of thyroglobulin, which is the inactivated thyroid hormone, what we have are these residues. So these thyroid residues, which are DIT or MIT, DIT standing for diiodothyronine, MIT standing for monoiodothyronine, and that just determines how many residues are being joined to form thyroglobulin. How many residues are being added to thyroglobulin to form T3 and T4, excuse me. So we have thyroglobulin, we can add DIT onto it. So if we add 2 DITs, that's 4 residues, so we have T4. If we add DIT and MIT, that's 3 thyroid residues, so we have T3. So that's the very slight or subtle difference between T3 and T4, how many of those thyroid residues, whether it was DIT, MIT, that were added to form our hormone, okay? So thyroglobulin, along with T3 or T4, again, depending on how many residues were joined there, is enveloped or engulfed into the cell via endocytosis, right? So that cell capturing via this vacuole or this vesicle that is moved through the cell toward the apical surface of the cell that is facing the blood. Sorry, so this is the basolateral surface, excuse me. This is the apical surface. So it's coming into the apical surface, moving down to the basolateral surface where it is going to be facing the blood, right? So the undersurface of this cell. Now, because this is a thyroid hormone, because it is a steroid hormone in terms of its origin, it does not need any help or receptors or transporters to cross the surface of the cell and enter the blood. It's going to go right through the membrane because it is a lipid-based molecule, and so it is going to go right through via diffusion. Okay, and so that is how we take our T3 or T4, move it into our bloodstream, and that is how we circulate thyroid hormone. Now, the action of thyroid hormone is basically lipophilic, so it acts like a steroid. That is what helps it to, again, move right through the cell membrane without any help. It's also going to alter the rate of transcription of mRNA. So it's going to change the rate, really important, change the rate at which we can make certain proteins and do certain things in our cells. That is how it's going to change the metabolic rate. We'll talk more about some of the details of the types of effects that it has, but essentially it's altering the rate at which our cells are functioning. Now, in the absence of adequate amounts of hormones available during fetal development, a disorder develops that's called cretinism. And cretinism is not only a physical deformity where there's usually this very specific affect and specific appearance deformity to the face, but there are also cognitive impairments as well. So they're usually learning disabilities that are associated with cretinism as well. So it's really important for pregnant women during that period of fetal development to have adequate amount of iodine, first of all, to make thyroid hormone, but also to have a healthy functioning thyroid so that that thyroid hormone can be secreted adequately. So the precursors to thyroid hormone, think about TRH from the hypothalamus, TSH from the anterior pituitary. If there's a problem with any of those steps in the process, we can develop cretinism in as a congenital deformity. Okay. Let's talk about the specific effects that this thyroid hormone has. So we said generally it has an influence on the rate of how our cells are making mRNA, are transcribing mRNA. But more generally, the types of effects that it has is called the calorigenic effect. In other words, these are the things in the cell that are going to cause more calories to be burned. It's going to be more of an increase in metabolic activity, more an increase in the rate of cell functioning. So it's going to increase the rate of sodium potassium pump activity. And we can think about this as more sodium being pumped out, more potassium being pumped in. And then we can also connect this to things like the resting membrane potential and the action potential. If my sodium potassium pump is pumping more sodium out more quickly, I can recover, so that refractory period, from an action potential more quickly, and I can generate a new action potential more quickly. So I can have faster rates of action potentials, in other words, faster cell functioning, faster neuron firing, and that type of thing, faster muscle contraction, etc. We also see increased number and size of mitochondria. So more mitochondria in larger size generate more energy, again, for more or faster cell functioning. We also see increase in oxidative phosphorylation enzymes. So similar idea, faster rates of cellular respiration, because we know oxidative phosphorylation takes place in the mitochondria, and that will also result in more energy faster. And then we see enhanced glycogenolysis, which is the breaking down of glycogen to make glucose, enhanced proteolysis, the breaking down of proteins to liberate amino acids, and enhanced lipolysis, the breaking down of fats to liberate triglycerides. The breaking down of triglycerides to liberate fatty acids. And so all of these things is basically liberating our sources of metabolizing these products in order to generate ATP, okay? So again, another way of expressing more energy production by the cell. We also see a promotion of gluconeogenesis and ketone synthesis, other ways that we can make ATP. And then we see a promotion of synthesis of beta-adrenergic receptors, which are essentially providing more opportunity for sympathetic innervation, right? So more of those adrenergic receptors, more norepinephrine, epinephrine combined, more rates of sympathetic influence. Okay, let us illustrate this axis here specific to the thyroid gland. So let's talk about how the thyroid hormone axis works and talk about how those other two hormones play a role in this axis. So we're looking at our thyroid hormone axis, and it's such a tongue twister for me to say thyroid hormone. I don't know, it's weird. So please apologize. So we're looking at the thyroid hormone axis. And we're starting with what we know is at the top of this axis, the hypothalamus. So about our hypothalamus here, communicating down. This is a bit bigger than I need it to be, so let's start over. So we're looking at our hypothalamus, communicating down to our pituitary, specifically the anterior pituitary, which is the larger part. We're also going to draw the posterior there, just for reference. So this region is our hypothalamus. Where we have this cluster of cells. This region is our anterior pituitary. And then we also want to think about the thyroid gland. So it's going to be further down in the body, right? Much more distant in terms of this axis here. Here's our thyroid gland. It's coming into contact with the trachea, as we know. So let's just draw the trachea here for a reference. All right. And this is the endocrine gland that is being signaled by the hormones that we are going to mention here in a bit. Oops. This is erasing everything except what I needed to. All right, so thyroid gland. All right, and then the tertiary organs, so other organs that are impacted by our thyroid hormone, we're also going to include those here in our axis as well. So things like the liver that are involved in all those processes we just mentioned, glycogenolysis, gluconeogenesis, proteolysis, etc., as well as the kidneys. Okay, so liver and kidneys. All right, so now we can start to add in our hormones and add in the direction of our axis. So TRH, thyroid releasing hormone, is being secreted from the hypothalamus. It's going to make its way down via the portal circulation that we described, and that is going to activate or enhance, so let's make it in green, that's going to activate or enhance the anterior pituitary to secrete thyroid stimulating hormone, or TSH, okay? So TRH from hypothalamus, TSH from anterior pituitary. Now from anterior pituitary, TSH is then going to be released into the blood, and that is going to stimulate the thyroid gland to make T3 and T4 by the process that we described earlier. So that is also stimulating thyroid gland to secrete T3 and T4. Now, T3 and T4 behave differently. We talked about a very subtle biochemical difference in terms of the makeup of these two compounds or these two hormones, but they behave very different. They're involved in sort of slightly two different processes. So T3 is actually more bioavailable. T3, and I'm going to kind of just use this arrow here. Hopefully I don't confuse you. Actually, let's put it on this side. So T3 is going to have an influence on the liver and kidneys more so than T4. So I'm going to put 2 plus signs here. It's going to really activate the liver and kidneys to do all of those things that we said before. T4, on the other hand, can be converted in peripheral tissues to T3. So T4 can be converted to T3, and then it will also have an impact on these cells. In other words, T4 does not have a role in terms of increasing metabolic activity directly to our cells, but T4 can become T3 that can have that more of an involved role, okay? T4, however, does play a more important role, which is the feedback mechanism. So we describe these 2 loops of feedback mechanisms, and this is where T4 comes into play. This is where T4 is more involved. So we're going to put T4 here again, and T4 plays more of a role in terms of that long loop feedback. So T4 goes all the way back up to the hypothalamus, and it's going to inhibit or turn off the secretion of thyroid releasing hormone, okay? And so this is going to be a long loop feedback. okay? And then we also describe a short loop negative feedback, and that is where TSH comes into play. So TSH also has this ability to go back up to the hypothalamus, to signal to the hypothalamus to decrease its secretion of TRH, all right? And this is an example of a short loop negative feedback. All right. And so really just to reiterate this flow here, we have TRH coming from the hypothalamus, stimulating TSH release from the anterior pituitary, stimulating thyroid hormone T3 and T4 release from the thyroid gland, the follicular cells here specifically. So let's put that here, follicular cells of the thyroid gland. And then that T3, T4 has two different roles, so to speak. So T3 plays more of an involved or hands-on role with the increase in metabolic activity. So think about liver, kidney, other types of target cells, increasing the sodium potassium pump rate, increasing those processes that we described earlier. Whereas T4 is more involved in the regulation of this. So if we need more of that T3, it can convert T4, it can be converted to T3. But if we need less, it can send the signal up to TRH to turn this whole thing off. So T4 is more involved in the regulation. Let's actually make a note of that up here. T3 is more involved in metabolic activity. So in regulating that metabolic in cells, whereas T4 is involved in regulation of thyroid hormone. Okay, and so we can convert T3 to T4 in order to send the signal that we need to shut it down, right? It's too much TRH coming, too much TSH. We can turn some of that T3 into T4, and we can send that as a means of regulating or having a negative feedback onto the higher parts of the axis here.